So it's start to have a presentation from the faculty member of HBP. So my name is Atsushi from the Faculty of Medicine, University of Tsukuba. Thus, my presentation is the sort of a side of the biology. So today I'd like to talk about the how the avian influenza adapt to humans and how the immune response restricts this adaptation process. So, as you may know, the influenza virus infects to respiratory epithelial cells, and upon this, the infection, the epithelial cells have a damage, and this finally induces the pro-inflammatory cytokine response, such as IL-1 beta. Then the IL-1 beta finally induces migration of the immune cells, such as the macrophages and the neutrophils. And these immune cells engulf the infected cells, so to remove lung. However, if these immune cells infect with influenza virus, these cells produce much amount of the cytokines. This will be finally induces the cytokine storm. So what we can say is the inflammatory response in the epithelial cells are crucial for the host defense mechanism as a first defender. So our purpose is to understand this cytokine response in epithelial cells. So this slide shows the schematic diagram of inflammasome in macrophages. The inflammasomes, this is a multimeric protein complex for caspase-1 activation. This caspase-1 is required for the IL-1 beta production. Inflammasome consists of the three proteins, that is the NLRP3, ASC, and caspase-1. It is known that the NLRP3, this protein functions as a sensor molecule to recognize virus infection. Then, upon virus infection, NLRP3 forms the homo-oligomers, and this NLRP3 recruits the ASC as an adapter molecule, and then this ASC further recruits caspase-1. Then this accumulation of caspase-1 finally forms the fully activated, proteolytically cleaved caspase-1. This activated caspase-1 cleaves from immature form IL-1 beta to form the IL-1 beta for its secretion. So far, all of these three protein molecules are known to be expressed in the macrophages. However, in the respiratory epithelial cells, there is no expression of NLRP3. But we know that the inflammatory response from the respiratory epithelial cells occurs. So this suggests that there should be unknown the sensor molecule instead of NLRP3. To identify this, recently we performed two types of the screening system. That one is the conventional interactome assay by using HE mass, and another one is the genetic screening using shRNA library for the whole human genome. From the HE mass interactome analysis, we identified more than 100 candidate proteins which bind to ASC only in epithelial cells. And we also found that 5 genes are responsible for the inflammatory response in epithelial cells. And then only one protein was identified from both screening. That was the MXA. MXA, this is a myxovirus resistance gene A. This is interferon inducible gene and a dynamic like large GTPase. Left panel indicates that crystal structures. From N-tymnus, this protein has the globular GTPase domain and the C-tymnus formed stop domain. This protein binds to ER membrane and forms the ring-like oligomers, which consists of the 16 constitutive MX dimers. MX gene is known to be a determinant of virus pathogenicity, but the exact molecular mechanism of this antivirus action remains unknown. So this slide indicates the alpha beta production in infected macrophages and infected epithelial cells. In the case of macrophages, the influenza virus infection induces the alpha beta production. When we examined the alpha beta production in the MXA knockdown cells, we did not find any reduction. But in the case of NLP3 knockdown, the dramatically reduced. This suggests that the NLP3 is responsible for the alpha beta production in macrophages. Then when we look at the epithelial cells, the NLP3 knockdown did not reduce the alpha beta production. This is due to there is no expression of NLP3 in epithelial cells. But the MXA knockdown decrease the alpha beta production. So in the case of epithelial cells, MXA is important for the alpha beta production. Then we overexpress the NLP3 in MXA knockdown cells. Then this reduction of alpha beta by MXA knockdown was cancelled by expressing NLP3. This suggests that genetically MXA and NLP3 have a similar function for alpha beta production. So next question is, what is the viral ligand which is recognized by MXA inflammasomes? To identify this, we performed immunoprecipitation assay from infected cells with anti-MXA antibody. LEN1 is input lysate, LEN2 is control IgG, LEN3 is anti-MXA antibody. Then we performed the Western blotting with anti-HA, M1, NP, and MXA antibodies respectively. HA is one of the glycoprotein, M1 is membrane scaffold protein, NP is the RNA binding protein which binds to the viral genome to form ribonucleoprotein complex, RNP complexes. Then from infected cells, we could successfully purify MXA protein and NP, but not other viral protein was co-immunoprecipitated with MXA. So this suggests that the MXA interact with NP, especially the viral RNP complexes. So next is the in vivo experiments. In mice, the MX1 is conserved as a homologue of MXA gene. But in the case of inbred mice, such as BAV and B6 mice, there is no expression of MX1 gene because of the large exon deletions. So the mice expressing human MXA gene we call human MXTG mice. Unfortunately, somehow the lines were disappeared, but the RNA shows alpha beta production. In the case of the non-TG mice, we could see around the 20 picograms per ml of IL-1 beta secretion in infected lung. But this IL-1 beta production was not observed in NLP3 knockout mice. This suggests that the NLP3 is important in the case of the non-TG mice. Then, in the case of MXTG mice, we could see around 4 times very high IL-1 beta production compared with non-TG mice. Then the panel B shows the migration level of the granulocytes. Here is neutrophils in infected lung. In the case of non-TG mice, the level of the neutrophils migration into the lung increased gradually along with the time course of the virus infection. In contrast, in the case of MXTG mice, the inflammation was rapidly activated day 1 post-infection, and that one was peaked at day 3. And then finally, that inflammation was calmed down at day 7 post-infection. So this suggests that MXTG mice produces very higher level of IL-1 beta at early phases of infection. So this one is survival curve, but the lines were also disappeared. But MXTG mice is survival well compared with non-TG mice. And that one was not observed in caspase-1 knockout mice. So this is mediated by inflammatory response. So same thing too for the NLP3 knockout mice. I'm sorry. Lines were disappeared, but in the case of NLP3 knockout mice, the MXTG mice was survived even in the absence of NLP3, so suggesting that MX gene is the important than that of the NLP3. So this is a summary. In macrophages, NLP3 recognizes virus infection, but in epithelial cells, MXA functions as a sensor molecule instead of the NLP3 to protect the virus spread into the lower respiratory tract from the upper respiratory tract. I may have still 1 or 2 minutes more. So anyway, this slide indicates the emergence of the pandemic viruses. Influenza virus, its natural host is aquatic birds. From the aquatic birds, virus transmit to intermediate hosts such as chickens and pigs, and then finally adapt to humans. Along with this, the adaptation process, the NP gene, one of the viral gene, is known to have adaptive mutation. Mutations function as adaptive mutation in the MXA. So MXA is thought to be an important factor to control the virus spread from the avian to humans. I'm sorry, I do not have time. Thank you very much for your kind attendance.